going quickly through just some other examples that relate to these kinds of ideas. So the recoded E. coli strain was an accomplishment and enabled a few different kinds of features, whether it be some improved virus resistance or the ability to incorporate non-standard amino acids. And the push along the way was to be able to do more writing of genomes using DNA synthesis. So rather than editing or engineering, although you could call this engineering, it certainly is, you would just synthesize DNA from scratch for chunks of or the entire genome. And so the next genome that people were really interested in was yeast. Much bigger genome, 12 million base pairs rather than 4. It's 16 chromosomes, as we discussed early in the class. So that makes it harder too. And so in this yeast project, which is still very much ongoing, people are maybe halfway, or at least what's published is halfway done. They're probably closer to 3/4 or maybe almost all the way done by now. They wanted to do a few things, showing that they could synthesize a yeast genome from scratch. They've taken out, synonymously substituted all of the UAG stop codons, and they've introduced in front of many genes the scrambling technology, which can essentially recombine different parts of the genome in an inducible way, so that you can randomly shuffle portions of the genome and genes that then give you this ability to very quickly generate diversity. But in also potentially a somewhat haphazard way. It remains to be seen how effective that will be. And so you can do this, as we talked about, yeast is very good at homologous recombination. So you can stitch regions of the genome in short regions together to assemble chunks, and then you can cross in chunks into the yeast chromosome and work your way towards a completely synthetic yeast. And so that's going on. Yeast is, of course, a very industrially relevant organism for metabolic engineering. And so it'll be interesting to see where that goes. In addition to that effort, there are also synthetic E. coli genomes underway, as well as reported, with less than 61 sense codons. So we talked about the stop codon reassignment effort. That effort was published in 2015. There's been more recent work towards both a 57 codon genome, as well as I think a 59 codon genome. And here, the idea is that you apply the same strategy to some of the sense codons. And once you start to do that, if you can successfully do that, it will have made a rather big impact, because in terms of virus resistance, now you are much more likely to have viruses that when they try to infect, and they'll probably succeed in at least the infection step of getting into a cell. Now, if in this case with an E. coli, if it's a bacteriophage, its bacteriophage genome or genes will probably have some of these sense codons, multiple in fact, such that even a mutation in 1 or 2 places by chance is not going to give you, the virus rather, the ability to make the statistical jump to have some kind of a protein that folds correctly. So this is a potentially powerful technology for that, and for this idea of if you wanted to use more than one non-standard amino acid at once. And so this is an ongoing project in the Church Lab, and there are lots to do here, because, you know, in some cases, for example, these arginine codons, just picking on this example, they're AG rich. In some cases, they are located within a gene, maybe towards the tail end, the C-terminal region, or I guess since we're talking about codons, the 3' region of a gene, where then in an operon, it might have been also dually purposed as an RBS. or have some other kind of secondary structure. And so hopefully, you know, as we talked about expression engineering, you can see some of the challenges that might exist trying to just change codons like this. Key point being, even though they encode the same amino acid, they're not necessarily the same at the RNA level. Here's the 59 codon E. coli. This was from Jason Chin's lab in Cambridge. I should have probably mentioned that the yeast genome project is an international effort spearheaded by Jeff Boki, who was in New York, but is now at Johns Hopkins. Or I might have it the other way. I think it's that way. But there are also many labs in China and Singapore and other parts of the world involved in that project. They've essentially split because it has 16 different chromosomes, although in principle you could do this with any chromosome. They've had different teams work on assembling different portions of the synthetic genome. So here, there was very recently published a 59 codon E. coli. Same idea, just taking 2 of the sense codons for serine and using synonymous substitution.